In this video, we're going to talk about Mendel, who is often described as being the father of genetics. What a claim. Mendel was an Austrian monk, and he lived from 1822 to 1884. And he was a monk, also a teacher, and quite an incredible scientist. He was really good because he did lots of replicates, and we'll look at some of the thousands of replicates he did. So lots of replicates. He took excellent notes of his work, so excellent and extensive note taking, and he also had a degree in statistics, so he did statistics on all of the things that he was identifying. So very scientifically robust work. And what he did is he crossed thousands of plants, and he could control the breeding of these plants. He did it with a paintbrush, and he went around mating different plants together with a paintbrush and then analysing the offspring. And he was working with the parental generations and the F1 and the F2 generations, as we discussed in the previous video. With his excellent notes, he came up with ideas such as inheritance, this idea of dominant and recessive traits. He identified that in his plant species. And he also was really good, like I said, at using statistics to support his studies. He was unrecognized in his lifetime, so he published his work. It was kind of largely ignored, rediscovered later, and then we realized how kind of academically robust he was. And like I said, particular strengths, replicates. We love a good replicate. He was very good at replicates. Statistics, and this idea that he controlled the reproduction. So he could decide which plants to mate together when he was studying the inheritance of different traits. The other key idea as well is that he looked at single traits one at a time. So he's looking at the colour of a pea, for example. Other geneticists at the time were trying to study things like human height and human intelligence, which even now we can't work out the genetic components of it because there are so many genes involved and so many factors, like lifestyle factors that affect it. He was looking at simple traits one at a time to study his work. And these are the 7 traits that he was really interested in. So he looked at the seeds, whether the seeds were roundish or wrinkled, and the colour of the seeds, whether they were yellow or green. The flowers, he looked at whether they were purple or white. The pods that the seeds were in, whether they were full or constricted, kind of like this shape. And whether they were yellow or green. And the position of the flowers, whether they grew in the middle, axial, or whether they grew at the very ends, terminal. And then he looked at the size of the plants. Were they tall plants or were they short plants? We now know that he was lucky that all these traits are simple, dominant and recessive trait inheritance pattern. But these are the traits that he was looking at. So we've seen this picture before, but what this is showing is one of Mendel's experiments, and he started off with his parental generation, the P generation, and what he did is he always started off with true breeding parents. We now know what this meant is that both of these plants were homozygous for their particular trait. So if we say, which we now know, that big P is dominant to little p, so this is purple and this is white. The true breeding means that the purple flowers in the parental generation were big P, and the white flowers in the parental generation were little p. And what he did is he mated those together, looked at the F1 generation, and kind of the really clever thing he did was this idea of a self-cross in the F1, so that he mated the F1 with itself, which he could do with plants. And remember, he was controlling this with a paintbrush. He then looked at what he's to see in the F2 generation, and hopefully you can start to see the number of replicates he's looking at. So he has got 705 F2 purple plants, and 224 F2 white plants. So quite incredible numbers of replicates. So this is why he had to keep meticulous notes, but also that's why his data is going to be really nice and statistically sound. So many replicates. This table summarizes the trait crosses he looked at. So we said there were 7 different traits, and we mentioned them all already. But we've got flower color. We've got flower color. We've got flower position, seed color, seed shape, shape of the pod, color of the pod, how tall the plant was. And what I want you to take home from this are the number of replicates in the F2 generation. So this is the F2 generation column. And look at these replicates. If you look at seed color, yellow and green seed color, 8,000 replicates in total. Eesh, crazy number of replicates. And so this is all the data from his F2 generation. And one of the things he noticed that in the F2 generation, he looked at the ratio of the dominant to the recessive traits. And he noticed that these dominant and recessive ratios are usually close to 3:1. And you'll notice some of them are further away, but statistically 3:1. So Mendel came up with a number of conclusions from his work. And remember, he wasn't acknowledged in his lifetime. But one of the things he came up with is he said there was no mixing of characteristics. We would use the term phenotype now, but there's no mixing of phenotype. By this we mean we have got purple flowers or we've got white flowers. We don't have codominance. We don't have purple and white spotty flowers. We don't have kind of a light purple colour if we had incomplete dominance. He was only looking at dominant and recessive inheritance patterns. He said that a characteristic could be present but hidden. So what he's identifying here is that you start off with a white colour, and in the F1 generation, the white has gone. So the characteristic is there but hidden. So we now know that the white colour is recessive, and in the F1 generation, you've actually got a heterozygous trait, and the dominant trait is winning and being expressed in the phenotype. He also said that phenotype is not the same as genotype. Okay, so that's we're using the terms phenotype and genotype now, but what he's identifying is the fact that these F1 purple flowers are different to the original parent purple flowers. And if we think about it, we said that the parents were always true breeding, like this, and the F1 generation are all heterozygous. So even though they both look purple, you've got this characteristic hidden. And this idea that the white flower is being hidden by the purple characteristic. The final thing he identified is this idea that the F2 ratio is always close to 3:1. And Mendel explained this by saying that each individual is inheriting 2 characteristics, or 2 versions of the characteristics inherited. We now use terms like genes. So when Mendel is talking about characteristics, we now would use the term genes, and the alternative forms, like he says here, we would use the terminology alleles for that. But absolutely a scientist before his time, and when his work was rediscovered, quite incredible that he had such meticulous data and such excellent statistical analysis. Thank you.